Hey, welcome back to another screencast. This one is going to be on T-cell activation. This is not an essay question, but the material covered within this screencast will show up on the exam and in a lot of different places. So it's stuff that you want to know, and it's a rather complicated process, so it's worth making a screencast for. So T-cell activation is going to be very similar, you will see, to B-cell activation in that you are going to get the formation of a clone, proliferation of selected cells, and then differentiation into active cells and memory cells. One of the differences is that T-cell activation is a two-step process where you have antigen presentation and co-stimulation. And then one of the other differences is that a T cell has the antigen, as it says here, presented to it by an antigen presenting cell, unlike the B cell that just bumps into the antigen with its receptor. Here we have an overview of the process. You have either your CD8 cell or your CD4 cell with their T cell receptor. And I should reiterate at this point in time that the T cell receptor is just like the B cell antibody or B cell receptor in that it is unique and is going to recognize specific antigens. So again, you have populations of individual T cells, be they CD4 or CD8, and when you get exposed to an antigen, your immune system needs to identify the individual cells within that population that have the receptor that can recognize the antigen so that they can then go out into the circulation and fight it. So you're going to get antigens presented to the T cells by the antigen presenting cells. This is going to activate the T cell. The T cell will then proliferate to form a clone. Most of that clone will go out and do whatever that type of T cell is supposed to do. Other members of that clone, be they CD8 or CD4, will remain behind as memory T cells, ready to mount a faster, larger response 3 years, 3 months down the road when you get exposed to that pathogen again. Let's start first with our antigen presenting cell. Here you have an APC. They present the antigens or display them attached to what is called the major histocompatibility complex, or MHC. So the purple thing with the little notch in the side is the MHC, and then the red thing is the antigen, which in this case looks like it's part of a virus that came from outside the cell. The antigen presenting cell is going to interact with the T cell. So the T cell, you already know, has on its surface the T cell receptor, which is unique to each T cell. It scans the antigen, just like antibodies bind and recognize antigens. And then it has this additional protein called a cell differentiation glycoprotein, which is responsible for docking with the antigen presenting cell. So when the two get together, it looks something like this. This is antigen presentation. You have the antigen presenting cell with the MHC and the antigen, and then the T cell has its CD protein docked into the notch on the side of the MHC. So the MHC serves as the docking port, if you will, for the CD proteins. So it's the combination of MHC and CD proteins that matches up the right cell with the right type of T cell, or the right antigen presenting cell with the right T cell. So the T cell receptor then scans the antigen. If this is something that is supposed to be in the body and everything is working well, this T cell will go about its business as if nothing ever happened. If this is a foreign particle and this T cell has the receptor that recognizes it, then this T cell, following co-stimulation, is going to become activated. Now you can have two different types of antigen presenting cells. You can have phagocytes. These are cells that phagocytize things. Two specific examples of phagocytes in your immune system would be macrophages which are the ones derived from monocytes, and then your dendritic cells. The other type of antigen presenting cell you would have is just a normal non-phagocytotic, possibly infected body cell. And the difference, as far as your immune system is concerned, between these two cell types is the type of major histocompatibility complex they use to present the antigens. Looking at the MHC, you can see there's two classes. All of your cells use class 1 MHC to present antigens, and then class 2 is reserved specifically for your antigen presenting cells, macrophages and dendritic sites. As we said before, the MHC is the docking port for the T cell CD proteins, and this is how the T cells interact with or recognize body cells, and it's how the T cell can tell the difference between a macrophage and an infected lung epithelial cell. MHC class 2 we'll do first. Class 2 MHC presents fragments of extracellular proteins that have been phagocytized or broken down in a phagolysosome. And these are going to be recognized by the CD4 protein on your helper T cells. So this just shows what the process of presenting antigens on MHC class 2 would look like. So you have a phagocyte of some sort, macrophage or dendritic cell. It is going to phagocytize a small particle, perhaps this is a virus. That virus gets digested when this phagosome merges with a lysosome. Then what is now called a phagolysosome is going to merge with a vesicle from the endoplasmic reticulum that contains your class 2 MHC, and the digested, no longer a threat to you and no longer a threat to this cell, these pieces, are complexed or attached to the class 2 MHC, and then this vesicle fuses with the cell membrane, and the digested viral particle or bacterial particle is now presented on the surface of this macrophage on the class 2 MHC for inspection by your helper T cells. So it is the CD4 protein on the helper T cell that is going to hook up with the class 2 MHC on a macrophage or dendritic cell. Class 1 MHC proteins are used to synthesize, are used to present proteins that have been synthesized in the cell itself. These are called endogenous proteins. If all is going well, then this intracellular endogenous protein is self. It is supposed to be there. It will not mount an immune response, or it will not provoke an immune response. If there is a virus inside of the cell, then this will be presented and recognized as non-self and will generate an immune response, or will act like an antigen. It is the CD8 protein on cytotoxic T cells that recognize the MHC class 1 protein, so that cytotoxic T cells are scanning your regular body cells. This is what presentation of proteins using MHC class 1 would look like. You have an endogenous protein, either part of the cell or part of a virus. Either way, it's floating around freely in the cytoplasm, not in a phagosome. Bits of it are chewed up by some intracellular machinery and then shipped over to the endoplasmic reticulum, where they are complexed with a class 1 MHC protein. This protein is then shuttled to the surface of the cell, and now you have class 1 holding out for the inspection of cytotoxic T cells, samples of whatever proteins are floating around inside the cell. So cytotoxic T cell is going to come over here. It's going to hook up to the class 1 MHC. So CD8 hooks into class 1 MHC. The T cell receptor scans this peptide, this small protein. If it is an antigen, indeed a foreign substance, then that cytotoxic T cell knows that this protein is not supposed to be here. This cell is most likely infected. This cell is going to die. Just to review real quick, you have antigen presenting cell presents the antigen on the MHC. The T cell docks with its CD protein, interfacing with the MHC, and then the T cell receptor scans the antigen. The second part that has to take place for this T cell to become fully activated is what's called co-stimulation, and that is the release of chemicals, which here they kind of look like pixie dust or something. But they're chemicals that the antigen presenting cell uses to fully activate the T cell. These chemicals that get released are interleukins. Interleukin 1 and interleukin 2 are the two most popular. And you can think of interleukins as the chemical language that white blood cells or leukocytes use to communicate. So interleukin in between white blood cell. Now that you have activation by the antigen presenting cell and co-stimulation, this T cell is ready to get down to business. It's going to proliferate, undergo many rounds of mitosis. It will form a clone army. Many members of that clone army will go on to do whatever they're supposed to do. So if these are helper T cells, these T cells are going to go further stimulate B cells. They're going to further stimulate cytotoxic T cells. They're going to release interleukin and really ramp up the immune response. And then this memory cell here is not going to do any of that. It is just going to wait along with, what should be a few thousand other memory cells, so that the next time you get infected, you've got 1,000 or so memory cells that already know how to fight the particular antigen that particular T cell receptor knows how to fight. So here's the quick overview one more time. You've got either your helper T cell or your cytotoxic T cell hanging out. You get infected. They get presented with antigens by the antigen presenting cell. In this case, they have dendritic cells on both sides, which I don't really get, but we won't think about that. The antigen presenting cell activates the CD4 or CD8 cell. There should be some pixie dust interleukin in here too for co-stimulation. Then you get full activation, proliferation to form a clone, and then most of the clone goes on to do whatever it's supposed to do, help or kill infected cells, and then you have memory cells left behind. And that is everything you need to know about T cell activation, CD4, CD8, MHC1, and MHC2. I hope you enjoyed it. Got it in just under 14 minutes.